Hi everyone, thank you for joining our webinar, Leveraging the Power of Global Datasets to Understand Aging in Bipolar Disorder. I'm excited to introduce our panelist, Dr. Lisa Eyler. Dr. Eyler received her undergraduate degree from Duke University and her PhD in clinical psychology from the University of Pennsylvania. She completed a clinical psychology internship and post-doctoral fellowship at the UC San Diego and the San Diego VA. Since 1999, she has been on the faculty of the Desert Pacific Mental Illness Research Education and Clinical Center, or the MIRECC, and since 2004 on the faculty of the UCSD Department of Psychiatry. She is currently co-director of the BioPsychoSocioCultural Unit of the MIRECC and professor of psychiatry at UCSD. She is also the director of the Center for Empathy and Compassion Training and Medical Education, a steering committee member of the UCSD Center for Healthy Aging, and chair of the Psychiatry Department's Chair Advisory Committee on Diversity Issues. Internationally, she is a member of the Older Adult Bipolar Disorder Task Force of the International Society for Bipolar Disorders, an investigator in several global consortia, including the Global Aging and Geriatric Experiments in Bipolar Database, the ENIGMA Bipolar Collaboration to share global neuroimaging data, and the International Consortium on Neurocognition in Bipolar Disorder. Dr. Eyler's research program is currently supported by several NIH grants on which she is an investigator. Thank you so much for joining us today, Dr. Eyler. Before you get started, I just wanted to remind everyone to please send any questions you have in the chat, and we'll get to as many as possible at the end of the webinar. Thank you.
Great. Thank you so much for inviting me. It's my pleasure to be here today to talk about some of the work that I'm doing. I'm going to share my screen. Okay, great. So thanks again, and thank you for watching this webinar. So I'm going to start by talking a little bit about the general issue of aging and bipolar, which is what I've been interested in studying for most of my career. And I am then going to speak about global consortia, bringing together people from across the world in order to better study what happens in bipolar disorder, and in my case, understand what's happening with aging and bipolar disorder. And there are several strengths as well as challenges of doing this kind of work that brings together people internationally to tackle important scientific problems. And the two consortia that I'm going to spend some time talking about are the GAGE-BD, which is looking at symptoms of bipolar disorder in late life and what happens in older people who have bipolar disorder when they get, in terms of their symptoms, and another group that I'm a part of called ENIGMA, and an analysis that I undertook looking at how the brain ages and whether the rate of brain aging may be different in people with bipolar disorder and what kinds of factors may influence the rate of brain aging. So starting here, obviously, folks on this webinar are probably very familiar that bipolar disorder affects 0.5 to 1% of the population, but maybe you didn't know that that was the case over the age of 60, that those were the prevalence numbers. And it's important because the world's population is getting older. And so it's important that we understand, are there some unique needs of older adults with bipolar disorder, which we sometimes refer to as OABD. We are wondering whether there should be age-specific treatment guidelines, for example, and also wondering from the standpoint of understanding biologically what's happening in bipolar disorder, could there be interaction of the normal biological processes that happen with aging with what's going on to cause the symptoms of bipolar disorder? A third important thing is that we know that there is both premature morbidity and mortality. That is, that people with bipolar disorder tend to die of all causes, but including natural causes, earlier than people without bipolar. and that people with bipolar tend to get diseases of aging at an earlier age than people without bipolar. So since these diseases of aging are seen in younger people, for example, cardiovascular, it suggests that there might be kind of a premature or accelerated aging process. And I'm particularly interested to see how that plays out in the brain and how it interacts with symptoms, including cognitive symptoms. And so seeing this has really led investigators to ask, is there an altered course of aging in bipolar? And if so, what might that look like? So one possibility is there's something like what you might call premature or advanced aging. The idea there being, if you look here in the slide at the blue line, you would see that compared to typical aging in the black line, premature aging follows a similar course, but it may happen sort of sooner in life. And so that if, for example, this dotted line indicates where the kind of impairment in brain or body would lead people to have an impact on their lives, you can see that in typical aging, you might reach that level of functional impairment at a later age, but with premature aging, you would reach that level earlier. Another possibility is that there's accelerated aging, and that means that the rate is actually faster, the rate of decline is faster. And one of the goals of work that we're undertaking is to understand whether there's accelerated aging or premature aging, whether this is the case in all people with bipolar disorder or just some, and what are the factors that might influence this trajectory of aging in people with bipolar disorder. And one of the factors that people have talked about is the idea that this premature or accelerated aging could be the result of the symptoms that people have. So the idea there would be that the longer you have symptoms of the illness, the more you will show a pattern of accelerated or advanced aging. And that's the idea of kind of progression, or in the case of the brain, people have talked about neuroprogression, that across the course of the disorder, things get worse and worse due to symptoms of the disorder. And those things are very difficult to disentangle, but it's something that the field is trying to start to tackle. Unfortunately, the evidence is limited so far about which of these trajectories might be the case and who might be susceptible to this kind of premature or advanced aging. One of the reasons that the evidence is limited is because there have not been very many longitudinal studies. Longitudinal studies being ones where you follow the same people over time. And part of this is because we're talking about following the same people over a long time period in order to really see these trajectories. So following the same folks for, say, 25 to 30 years. And that's very difficult to do. Also, most of the work that has been done at individual sites around the globe, and most of those samples are pretty small, partly because, as I said before, people with bipolar disorder unfortunately die sooner, and so there are fewer older age bipolar patients to work with. Also, the fact that not every person would like to be involved in research studies, and so the samples are generally small. And when you get lots of small samples, you oftentimes get mixed results. Some studies find one thing, other studies find another. And partly that may be because the samples that they are drawing are different. So for example, you might have a group of people who have had the disorder for a very long time. You might have another group of people where most of them have just more recently been diagnosed with bipolar disorder. You may have a different gender distribution, et cetera. So the fact that the samples vary in their composition makes it difficult to get the same results every time from studies. And another thing that makes it difficult is that people use different methods and they use different measures across their studies. So what's really ideal would be a multi-site prospective longitudinal study. So what does that all mean? Multi-site would be lots of different sites, of course, different representing different parts of the world, for example, different parts of the country within the US. Prospective means start from the beginning, follow people when they're young or when they're first diagnosed. And then longitudinal means to follow those same people over time and get a sense of each person's individual trajectory. The problem is that these kind of multi-site prospective longitudinal studies are very expensive. It means that you have to coordinate across a bunch of different studies, and you have to give people incentive to stay in the study for the long run. They're also difficult to do with normal kind of national grant funding, which the typical cycle for grants in the US, for example, is 5 years at a time. So to say, I want to follow people for 25 years, it might be pretty difficult to get that done because every 5 years you have to reapply for more funding. And they're very tricky to coordinate. Luckily, there are some efforts afoot right now to design some global bipolar longitudinal studies using money donated from philanthropists and others, and I'm hopeful that those will get off the ground. In the meantime, what we do have is a lot of studies that have already been done across the country and across the world that have data that we think would be interesting to combine together and integrate so that we could ask important questions. So this is where the idea of a global consortium comes in. And I'm involved in actually 3 different global consortia on the topic of bipolar disorder. And they all have a similar format in that the idea is we combine data from multiple existing studies of bipolar disorder. These individual studies could be cross-sectional, meaning they just measure people at one point in time. They could be longitudinal, meaning they follow people over time. They could be interventional, meaning that they're studying some kind of a drug or psychosocial intervention. And whatever the case may be, or they may actually just be data that are collected in the course of routine clinical care. So some studies, some countries, for example, have databases of all of the patients that are seen clinically that can be used to contribute to this kind of work. And then from these global consortia, we can do different types of analyses in order to combine data. One possibility is something called a meta-analysis, which you may have heard of before. It's a way of taking the results from individual studies and combining them together to see what the overall result seems to be. And there, what you do is the individual investigator does all of the analysis of the data and says, this is what I found. I found that this part of the brain was smaller in people with bipolar disorder than in people without by about this much. And then you take that information from every study and you combine it together to say, on average, the difference was found to be this across studies. Another type of analysis that's even more powerful, though, is what's called a mega-analysis. And a mega-analysis means that every study and every site actually submits the individual data from the patient participants and the healthy comparison participants from their site, so that the investigator that's putting it all together has a lot more samples, a lot more individuals in their analysis. And instead of looking at just the overall results of each study, you're actually getting the individual person's data. These types of analyses are very powerful, as I said, but they also lend some difficulties in terms of privacy and confidentiality. And some countries are have rules that tend to prohibit the sharing of individual participant data. Another issue with the global consortia, if you are going to do a mega analysis, is that you want to have harmonization, and you want to have harmonization of 2 things. One is harmonization of the processing methods. So if you have a certain type of data, but everybody processes that data in a different way. So if, for example, if you have brain images and brain imaging data, there are many different programs, software programs, that allow you to do things like measure the size and shape of different regions of the brain. If everyone uses a different processing method, it will be very difficult to combine the output because you might get the output in a slightly different format for each of those methods. So having some standard way of processing the data at each site is important for these type of consortia. And another thing that's important is harmonization of the measures. As I mentioned before, one of the things that makes it difficult to see consistent results across studies is that every study may use a different measure. So for example, some studies might use a rating scale for depression called the Hamilton Depression Rating Scale. Other studies might use something called the Montgomery-Asberger Depression Rating Scale. So the results from that are on a different scale. You know, they go from say 0 to 20 versus 0 to 50. So how do you bring data from those two instruments together to understand depression severity? And we're going to take a look at how we did that in one of these global consortia studies. So in general, the strengths of having these global consortia are their power to detect smaller effects than you would be able to see with individual samples. Oftentimes you will have thousands of individual data points in a global consortium data set, as opposed to the usual 100 or less than 100 in smaller studies. So we're able to find things in those big data sets that we might not be able to observe in the smaller data sets. It also allows us to do some newer types of analysis, like things called machine learning. You may have heard about this, where you use the data and you explore the data in a way that's not able to be done if you only have, say, 100 subjects, but having thousands of them. And I'm going to talk about a particular kind of machine learning that we were applying in our data set. And the other strength of a global consortium is the representativeness. We always in science want to have the sample that we are studying be as representative as possible of the population that we're trying to make draw conclusions about. And in the case of a global consortium, it allows us to have representatives of how bipolar disorder manifests across the globe. Whereas each individual study generally only has participants that are living in that particular region. And finally, it gives us the opportunity to even ask questions about, are there differences in the way that bipolar disorder manifests across different countries and in different regions? So that once we've put all the data together, we can then go back and say, what were the differences between the sample that came from Africa versus the sample that came from the US, or that came from Australia versus the sample that came from Europe? So we're able to look at those kinds of differences, and that's not something that you could do without a global consortium. However, there also are challenges, some of which I've already hinted at. One of the challenges is that every time you harmonize measures, it decreases the granularity. So what do I mean by that? What I mean is that if you had a scale that was on from 0 to 30, and then you had another scale that was from 0 to 25, generally when you try to harmonize those scales, you end up with a scale that's, say, from 1 to 4. So mild, medium, severe. Or absent. And so you end up with kind of more coarse data than you started out with when you tried to harmonize across measures. Also, because you didn't start from the beginning and say, let's build a consortium where everybody uses the same measures and everybody collects data on the same types of characteristics, you find that when you collect all the data from existing studies, everybody didn't measure the same things. So some people didn't measure education level, or some people didn't ask about how many times a patient was hospitalized, or maybe they didn't do any cognitive testing. So you do have limits in terms of the analysis that you can do, because not all studies measured the same thing. Another issue is that when you do find these small effects because you had a large sample, it raises the additional question of, are those small effects meaningful? Is there actually something that affects patients' lives from a difference of 10 milliliters between the hippocampal size of people with bipolar disorder and people without? And so it's really important for us to not only find these small effects, but understand whether those small effects add up to something that's clinically relevant and functionally meaningful. There is still a limit to the generalizability. So although we're getting more representation of patients from across the world, each of the studies that contributes to the global consortia had their own biases in terms of who decided to show up for research studies. And we know that obviously people who are very depressed, for example, or people who are homebound for the reasons because they have physical morbidity, or because they have a understandable mistrust of science, all of those keep people away from being in research studies, and that happens across the world. And so those biases are still in the data that we analyze. And then finally, as I mentioned before, too few of the data sets that already exist are longitudinal, where they follow people over time. Those are out there, and we're definitely interested in combining those data to see what we can see. But there are too few of them to really make a lot of inferences about the trajectories at this time. So I want to start by telling you about one of the global consortia that I'm part of, and this is the Global Aging and Geriatric Experiments in Bipolar Disorder Database Project, or as we like to call it, GAGE-BD. And the other PIs of this consortium along with me are Annemiek Dols from the Netherlands, Soham Rej from Canada, and Martha Sajatovic, who's also here in the US. And we have members from across the globe, and we're adding members weekly. This project is supported by the International Society for Bipolar Disorder and a particular research initiative called the Bowden-Massey Strategic Research Initiative. And so we're very thankful to Dr. Bowden and Ms. Massey for their support of this work. Our GAGE-BD project was conceived in 4 different stages. The first one was to create a data community and address regulatory issues of making this possible, standardize the data in formats that would be suitable for analysis, including some of the harmonization that I talked about, and then to investigate our main aims, which I'll talk about in a minute, and facilitate other investigators doing secondary analyses, and then establish some guidelines and infrastructure that would help us to perhaps engage in more prospective global studies, where we start from the beginning working together and collect new data all together in the same way. As of right now, we have 20 contributing data sets, and you can see the dots on the map show the areas that we are getting data sets from. And I'll point out that, of course, we do not have a complete coverage of the map, and there's a lot of coverage that is in North America, for example. And we're working on that in our second wave to increase the number of sites that are participating and increase the global representation. So our first stage has been completed. We got IRB, the Institutional Review Board requirements were met for any of the local IRBs that contributed studies. The lawyers got together and figured out ways of them sharing the data between institutions using these data use agreements. We had all of our investigators sign a memorandum of understanding about how they would publish or present on the data. And we secured a set of data, uploaded a set of data, as well as a data dictionary, which describes what the data are and how it was collected. And we had that, we created a lock of the data, meaning we weren't going to look at anything that came in after that for our analysis in March of this past year. And then when we brought the studies in, we looked to see what were the variables that they measured, what were the characteristics, and also what were the characteristics of the studies, the original studies, like were they clinical trials, et cetera. So stage 2 has also been completed for our study. And these are some of the descriptives of the variables that we have. So what you can see is that some variables were easy to integrate together. For example, age, age of onset, and almost all of the studies used the Young Mania Rating Scale to measure severity. So you can see in this table how many studies had each of these variables, what the number of participants was overall when you combined all those studies, and then something about the characteristics of this global sample. Some things we did have to recode. For example, people coded gender in different ways. They might have used 1 and 2 versus 0 and 1, or M and F, or some would have a non-binary category. So we did have to do some recoding of the variables for those types of things. And then there were some that we actually needed to harmonize. So for example, for years of education, some of the data sets may have sort of said what level of education they got to, and we had to convert those into years so that we could have years of education for everyone. And then for depression severity, as I mentioned before, we had the Hamilton and the Montgomery-Åsberg, and those two we were able to put together into depression severity bands: absent, mild, moderate, and severe. And then we decided to do some analyses of our initial aims. And first, we just wanted to see how do symptoms seem to differ across the adult lifespan. Are there differences in the severity of depression or mania in older versus younger patients with bipolar disorder? And then see if there are any particular factors that seemed to have a different that seemed to lead to a different relationship with age. So we looked to see whether people who were more educated seemed to have less of a downward slope or less of a change with age in their symptoms. But again, keeping in mind that these were all cross-sectional data. So these were the baseline data from any longitudinal studies. It was just a one-time snapshot. So what we looked at first was whether there were any sample effects. And we tried to see, you can see here that the different samples had different age ranges. So this is a distribution of age for each of the different samples, the 20 different samples. And so you can see that most of them had kind of a mean age that was over 60. That was sort of between 55 and 75. There were some studies where the mean age was a little bit younger, but they did include people over the age of 50. And then there was this study that had a very broad age range that was participating. And so we tested our different hypotheses. We expected that the older people in our sample would have less severe mania, but more severe depression. And that was based on some previous work with individual samples. And then, as I said, we were interested to see what would happen with if there were differences between men and women, or between more highly educated individuals and those with less education in terms of how age related to symptoms. So our two main results so far was actually that, consistent with what we had expected, we did find that the older participants in our study had less severe mania than the younger participants. So here is age on this axis. So these are our older participants up here. And in general, and this is the Young Mania Rating Scale, which is mania severity. And so you can see that in general, those that are older have slightly less severe symptoms of mania, whereas the younger participants down here have more severe mania symptoms. We also, however, contrary to our expectation, found less severe depression in older bipolar participants as well, as opposed to more severe, which is what we had expected. So once again, our older participants are in the lower depression severity group. So this is absent, mild, moderate, and severe. So you can see that there's more, it's an older group that's in this lowest severity category. What factors seem to be related to to symptoms and to the aging pattern and symptoms in our study? Well, first of all, we found that men and women in our study did not differ in their symptom severity. So the mania rating scores were similar for men and women, as were the depression rating scores. And similarly, both men and women showed the pattern that I just showed you overall, which is that older people had less mania severity and less depression severity. In addition, we found that education was not related to symptom severity. However, we did find that, that those who had less education seemed to actually show even stronger an even stronger trend for there to be lowered depression as you got older. So those with less education showed more of this age decrease in depression severity. So our conclusions from this initial analysis is that we could successfully create a global collaboration to create this large integrated data set for testing our hypotheses in older age bipolar, and that there was a lot of differences among our samples, but we could still combine them and find some interesting results, including that our older patients seem to have lower depression severity and lower mania than the younger patients in our sample. So it could be, because these were cross-sectional data, it could be that those with most severe symptoms were not in our data set, particularly those that were older, who may not have made it to that late age to be in our research studies, or who may have been too severe to come in and do research. So there's definitely problems, as I mentioned before, with cross-sectional data. So future directions for this are that we would like to test our additional aims having to do with functioning and medical comorbidity, and that we are inviting our other contributors to think of new analyses that could be done, including on gender differences, analysis of differences based on what age the symptoms of bipolar started, differences having to do with people who have more versus less medical comorbidity. And there also is an analysis looking at cognitive data for the sites that do have cognitive data. We also have opened up for a second round of data collection, and we're going to be welcoming in at least another 10 or 12 sites to our data set and our consortium to help us to do that. And we're going to be also inviting sites who have neuroimaging data to contribute those. So that's the GAGE-BD project. Another consortium that I've been involved in is the ENIGMA BD working group. So this is enhancing neuroimaging and genetics through meta-analysis project. And this is the Bipolar Disorder Working Group. And as you can see here, there's also a global representation of cohorts, again, with a lot of concentration in Europe and North America. And we're also making attempts to have better representation globally in ENIGMA. This is made difficult by the fact that you need a lot of resources to do neuroimaging studies, and you can't just get, for example, clinical data. It has to be a site that has an MRI machine. And the goal of ENIGMA is to allow individual investigators to process brain imaging data in a standardized way so that it can be shared and combined together to do global analysis. And there are a lot of challenges to neuroimaging work in general in bipolar disorder. There's a lot of variability among patients, and this can affect the results of neuroimaging studies. There is, as mentioned, a very high cost to doing MRI research. And so by taking advantage and combining across smaller studies, we can have more power. As I mentioned, people tend to process their data in lots of different ways, and this group has come together to standardize how you process the data in order to get out the same measures for every contributing site. Again, powerful, these combined analyses are more powerful, allowing you to detect smaller effects than for individual studies. And again, mostly, we are looking at cross-sectional studies, but there's a growing number of longitudinal studies that we can also put together. And we have worked together to decide, how to combine and how to work together in terms of making hypotheses and publishing the results of our study, whether they come out positive or negatively. That is, whether our hypotheses were confirmed or not. So what are some of the things that the ENIGMA Bipolar Disorder Consortium have found so far? Well, this study shows that the parts of the brain that are in the middle of the brain, including things like the hippocampus, which is important for laying down new memories, the amygdala, which is important for fear responses and other emotional responses, the thalamus, which is an important relay station in the middle of the brain, as well as the ventricles, which are the CSF-filled fluid spaces, which tend to grow bigger as you get older. You can see that across 1,700 bipolar participants and over 2,500 healthy comparison participants, there was evidence for significant reductions in the size of the amygdala and hippocampus and thalamus, and increase in those fluid-filled spaces or ventricles. And so we found that this was the case when we combined all of these data. Another set of studies looked at the surface of the brain, or the cortex, and examined how thick or thin that gray matter strip is, where the neurons, the bodies of the neurons reside. And here we were able to show that in this picture, you see that there are regions in the kind of warm colors where the thickness is lower. So there's a thinner cortex in people with bipolar disorder than in people without bipolar disorder in these areas that are kind of in orange. And really didn't find any evidence of regions that were thicker. I was interested in aging of the brain, and those data suggest that the brains of people of many different ages all put together look a lot like what we would see in older people without bipolar disorder, in that in older people without bipolar disorder, we certainly see a shrinkage of those subcortical regions and an increase in volume of the ventricles, and we often see thinning of the cortex. So it kind of looked like that bipolar disorder had an older looking brain overall than people without bipolar disorder. But we wanted to examine that more specifically. And so I undertook a secondary analysis of the data from the ENIGMA Bipolar Disorder Working Group, looking at something called brain age. So again, we know that there are differences in outcomes in terms of aging, differences in longevity in bipolar disorder that suggest that people's biological age and their chronological age are a different thing. And we know that different biological systems can age at different rates. So it's important for us to think about how we can determine what someone's brain age is. How robust is that person's brain compared to other people of the same age? And that can help us maybe understand differences between people in things like how well they are doing cognitive tasks, what their symptoms are, et cetera, as they get older. And so conceptually, what brain age does is that it takes a very large sample of people without bipolar disorder to find a combination of brain features which accurately predicts your chronological age. So from all the different things that we know about the brain from a brain image, how can we find a combination of those that together predicts how old you are? Now, this seems like kind of a silly thing to do because you can just ask somebody how old they are. But again, what we're trying to understand is what combination of brain features is most related to somebody's chronological age, so that we can then apply that algorithm to a new sample, perhaps people who have a psychiatric disorder like bipolar disorder. And then what we do is we apply the algorithm and we get the age that their brain predicts that they are. And then we know their actual age. And so we calculate the difference between those two. And that's what we sometimes call the brain predicted age difference. So basically you could be 50, but you could have the brain of a 65-year-old. And so you would have a 15-year brain predicted age difference. And there have been studies that have looked at brain age, smaller studies looking at brain age that didn't see any difference in this gap in people with bipolar compared to those who did not. And you can see the sample sizes for those studies here. However, as I mentioned before, a lot of these big studies did show that the brains looked older, but nobody has specifically looked at the brain age gap in these large samples. And so we took a model that was developed through a different working group to predict chronological age in their healthy control data set, healthy comparison data set, and we applied that model to our bipolar and our comparison group. And our group had 599 men, 929 women with bipolar, and 646 men and 757 women who were healthy comparison, non-mentally ill participants. And we had 25 sites that contributed data for the bipolar group and 23 for the comparison group. And what we found is that this model predicted pretty well. So you can see that this is the brain age prediction, and this is their actual age. So you can see that for the men and for the women that were, the models were separate. They had pretty good prediction for our normal non-mentally ill comparison group, and for our bipolar groups, we also saw pretty good fit, maybe slightly worse fit in the women with bipolar disorder. And then we compared the difference between their predicted age and their actual age. And what you can see here is on the left side, there's not much of a difference in the healthy comparison folks, and there is a small but slight difference in the bipolar group. When we did the statistics for this, it was highly significant, but it was only about 2 years greater gap in the bipolar group. But interestingly, then when we looked at what factors predicted who had a big gap, so whose brain looked a lot older than it should, versus those that didn't have a brain that looked older than it should, or even had a brain that looked younger than you would think for their age, we found that people who had bipolar 1 compared to those who had bipolar 2 had a bigger gap. We also found that those who were taking anti-epileptics or antipsychotics at the time of the scan had a bigger gap, and those who had started there with the symptoms of bipolar disorder later in life also had brains that looked older than they should for their age in general. Interestingly, people who were on lithium at the time of the scan had a lower brain brain gap, the lower difference. So lithium seemed to have been kind of protective, and there didn't seem to be any relationship with this gap for whether or not you were euthymic at the time of the scan, whether patients were on antidepressants, or whether or not they had psychotic features as part of their illness. So again, this is just an example of the neuroprotective effect of lithium. So people who were on lithium at the time of the scan had a much, had a lower gap. Their brains actually looked younger than they would have been predicted to look given their age. So in summary from this study, this large-scale study revealed some small but significant differences in this gap between your predicted brain age and your actual age. And the differences among people in the degree to which the brain looked older than it should seem to be related to features such as lithium use, for example. And we're working on expanding kind of the training sample and being able to look at more fine-grained brain features in the coming years. So finally, I just wanted to wrap up by saying that there things that we still need to know about the aging pattern in people with bipolar disorder is that we really still need to have these long-term longitudinal trajectory studies to understand what happens with symptoms, cognition, and the brain in individual patients over time. We also want to understand what makes different trajectories happen, and what can we predict early on that allows us to know if somebody's at risk for accelerated brain aging. And therefore, what are some of the modifiable risk factors that we might be able to attack in order to prevent or slow those declines in people who are vulnerable? So thank you so much for your attention, and I'll be happy to take any questions that you might have.
Thank you so much, Dr. Eyler, for that wonderful presentation and for sharing your research with us. It looks like we have time for a little Q&A, so I'm going to get started with some of the questions. So our first question is, someone asked, is it possible that mania and depression symptoms decrease with age because individuals have learned how to cope with their bipolar symptoms?
Absolutely. I think that that's a very important possible explanation for those decreases. And I would love if that was the case, because it shows that there are ways that people can manage their symptoms so that they're less functionally impairing, and that people can figure out ways to treat them either pharmacologically or with psychosocial interventions that work for them. And so that very well could be the case for those results.
Okay, thank you. And then the next question, somebody wanted to know, is it safe to say that individuals with bipolar disorder can likely experience a decrease in symptom severity as they age?
Yeah, so again, I would love to say that is the case, but again, our data right now are cross-sectional, so it would be easier for me to say yes to that if we had more data that had followed the same people over time, because again, it could just be that our older sample is different in some way, and that they have, for example, always had less symptoms than the younger sample that we studied, and we've just caught them as when they're older, but they've always been less severe for some reason. And so it's hard for me to for sure say that symptoms will reduce. However, I think the first question leads us to have some optimism that people who learn strategies for coping with their symptoms, who figure out through what I know is a often painful trial and error process, what kind of treatments work for you, that there definitely is the hope that individuals will see a lessening of their symptoms as they get older.
Okay, thank you for that explanation. And then lastly, someone wanted to know, how can individuals who want to participate in a study like similar to the ones you talked about get involved?
Great. That's a wonderful question. And thank you, because that's definitely something that you can do to help. So generally, the studies, particularly the ones that are brain imaging studies, will be carried out at local academic medical centers in your area. So going to the websites of the departments of psychiatry for your academic medical center will often show different ways that you can get involved in research studies. And similarly, I know that the International Bipolar Foundation in their newsletter oftentimes sends out announcements about research studies that you can participate in, either somewhere locally or um, through virtual contexts, or online surveys, et cetera. And so it would be great to have as many people as possible, particularly groups that typically don't participate in research. The more that we can have a representation from all types of people in our studies, the better.
Okay, great. Thank you so much for answering those few questions. I just want to remind everyone, if you want to rewatch this, you can find it on our YouTube. And again, thank you so much, Dr. Eyler, for this wonderful presentation.
You're very welcome. Thanks everyone for being here.